This session is Virginia Miller from Mayo Clinic, and she'll talk to us about, are there sex differences in small vessel diameter? I'm sorry, small vessel disease. Sorry.
Okay, thank you very much for the opportunity to talk to this group today. And where's Jerry? Thank you, Jerry, for setting me up with this topic. And I've asked a question, and I think you probably already know the answer. And I'm also going to sort of give you a high-level talk of some concepts you already know. And I'm not saying these things to insult you, but I'm saying them to remind you to keep them in the forefront of your mind as you consider the systems that you're investigating, because sometimes these concepts tend to get put to the side. So here are my disclosures in terms of funding, and thank you very much to the National Institutes of Health who are keeping us going. And I have three very simple objectives in this 15-minute talk today. First of all, to define how sex is an important biological variable. Second, to list some points of investigation of mechanisms into sex differences. And finally, to paint the big picture. And as I go through these today, I think you're going to see some repetitions and themes. Already, I'm hearing the same kinds of words and concepts that are infiltrating, and I think you'll hear that through the rest of the presentations today. So what are sex differences and what are sex biasing factors? We're talking about sex here today, not gender. Sex is what? Biology. The basic presence of an X and Y chromosome, the sex chromosomes, or an XX chromosome in females, and the reproductive organs that result from that in the hormonal environment. Gender is everything else. It has an impact on what these genetic processes might be. But for today, we're focusing on biology, that is sex. So in males, we have an X and Y chromosome. In females, we have two X's. And when I gave this to my group, they said, gee, Virginia, you need to bold the other X. And I said, no, I don't, because one X, to consider the dose of the X chromosome, is inactivated. So if you have genetic polymorphisms on the X chromosome, they're going to show up in the male, but maybe not so in the female. Now, we used to think that the mosaicism in the presentation or inactivation of X was random. We're now considering this that this may not be as random as once thought, and I'm not going to dwell on that today. The presence of the X and Y chromosome in every cell dictates the nature of that cell. But on top of that, and even structural organization of some of the organs prior to the development of the sex organs and chromosomes. However, on top of these chromosomes, you have the sex hormones. And let me remind you that both testosterone and estrogen are present in males and females. Aromatase, the presence in some organs, the heart and the brain, for local production and conversion of estrogen in men is becoming quite important, but also to keep in mind that the relative concentrations of these in the body varies over the lifespan, which I'm also supposed to talk today about aging. So what you have then is a sex biasing factor that varies across the lifespan. So we consider the cardiovascular system of men and women. It's a given that they shouldn't be expected to act the same, because as you can see here, a female's body is programmed, the circulatory system is programmed to be able to adapt to this very complicated situation, physiological process, which we call pregnancy, which involves adaptation of the circulatory system not only to maintain the health of the mother, but also to maintain the health of the fetus. So when we're talking about basic molecular mechanisms which contribute to health and disease, is a given that these systems may not be the same in men versus women and vary across the lifespan given changes in the hormonal environment. So we need to take an integrative approach to how we consider sex in terms of a contributor to cardiovascular disease. It's going to affect the brain and neural control of the circulatory system, the organs that contribute to the heart to circulate the blood, the kidney which help regulates it and clears the blood, and also all components of the blood vessel wall, including those elements which circulate within the blood. As we go forward today, we need to remember that every cell within this whole system has a sex, and that the hormonal variation that you're going to see affecting the expression of these genes varies across the lifespan. So how does this play out? Well, let's consider some conditions which may have sex differences: cardiovascular disease and incidence and presentation, chronic kidney disease, vascular dementia, ischemic, non-obstructive ischemic heart disease, all have sex differences in either incidence or presentation. But we also have sex-specific conditions, which the first speaker mentioned in terms of erectile dysfunction and preeclampsia in women. The sex-specific cardiovascular conditions usually are associated with reproduction. But the question is, how do these sex-specific conditions influence the sex differences across the lifespan? If these sex-specific conditions influence the vasculature, the blood elements, the endothelium, the smooth muscle cells, and the parasites, which were mentioned in the previous talk, then how do they affect organ damage in the heart, brain, kidney by changing blood flow, altering structure and function across the lifespan? To give an example of how this might play out, let's look at the sex-specific condition preeclampsia in women. With my colleagues Vesna Garovic and Michelle Milkey, looking at the Rochester Epidemiology Project, which is a medical record link system where we're able to evaluate what's documented in the medical record in the population that lives in Olmsted County. They evaluated hypertensive pregnancy disorders relative to future cardiovascular disease. So these were women who gave birth between 1976 and 1982, and we're now pulling their records to see what happened to them in the intervening years, but they're now in their mid-50s. The hypertensive pregnancy disorders were defined as preeclampsia, and these were medical codes that were pulled from the records. Those women many years later had a significant increase in hypertension, a significant increase in reported dementia and stroke, but not coronary heart disease. You might say that this is because these women were too young to have coronary heart disease, or that the codes may not have differentiated the particular types of coronary heart disease, or more importantly, there might be different etiologies between the two. And I see one of our pregnant ladies here in the front who's shaking her head. Okay, so health to you. Okay, so I want to go back. Let's see. Why can't I go back? There we go. There, okay. So related to hypertension, future hypertension, we also see two conditions which affect the brain. Well, let's look at how this might be manifested in the brain in terms of small vessel disease, and using structural MRI, magnetic resonance imaging of white matter hyperintensities, we're using this as a surrogate for small vessel disease in the brain. So this is a horizontal image through the brain. The nose is up here, the back of the head is here. Here's normal brain tissue, and you can see nice myelin sheaths and healthy cells, where if you look at this area called considered white matter hyperintensity, which is a damaged area or ischemic area, we see loss of myelin and also loss of cells, including capillary density. What happens to these women as they age? Well, we used another condition specifically to women to evaluate this in a prospective manner, and that is the other female-specific condition of menopause, of loss of hormones in midlife. And in the Cronus early estrogen prevention study, we imaged women between the ages of 42 and 58. The mean age was 52. Again, we have this horizontal look through the brain. The white matter hyperintensities here are masked so that they can be better quantified in green. The average age of the women that we investigated in this study or examined was 52. I consider this pretty young relative to aging. And what is amazing to us was that every woman we imaged within this age group had white matter hyperintensities. And as we followed them over the 4 years, these increased, such that we saw a linear progression of the white matter hyperintensities over the 4 years of the study. The other amazing thing within this study of the early estrogen, Cronus early estrogen prevention study, was that these women were screened for their pre-existing cardiovascular risk factors all which were within the normative range. These were women at low risk for cardiovascular disease and had no calcium in their coronary arteries. So this was pretty remarkable to us that this starts early. It's well known that mean arterial blood pressure may increase the risk for development of white matter hyperintensities. But when we evaluated the mean arterial blood pressure in these women, the remarkable thing was that we did indeed see an age-related increase that the white matter hyperintensities increased with increasing blood pressure, mean arterial blood pressure. But look where we are. These mean arterial blood pressures is not what you're going to be considered as hypertensive. So we have a relationship of blood pressure as a continuous variable with the development of white matter hyperintensities in the brain. These were adjusted for age. What does this age adjustment look like? Well, if we break it out, we see that the women who were in the middle 50s, here is their relationship to white matter hyperintensities and blood pressure for age 55. Here it is for the age 60. So indeed, there is an age interaction with blood pressure and development of white matter hyperintensities, but what seems to be remarkable is that it decreases with age over time. And so when we do statistical adjustments for some of these interactions, we may be missing a critical time point where intervening to slow progression of some of these diseases might be missed. Do these increases in white matter hyperintensities or blood pressure have any effect on end organ function? Well, if we look at the cognitive ability of these women using auditory attention and work and memory tests, we see that the only conventional cardiovascular risk factor that correlated with a deficit in these two cognitive parameters was blood pressure. Again, within a pretty normative range, indicating that the relationships between some of these peripheral systems and what's going on in the brain needs to be paid attention to, and particularly the critical time points within menopause or with individuals with hypertensive pregnancy disorders to be intervened. We heard some activity questions from Jerry Meininger regarding microvesicles, and I'd like to say that we can look beyond conventional cardiovascular risk factors for mechanisms that might contribute into some of these factors. The one that we were particularly interested in are activated platelets, because platelets have been implicated in the development of cardiovascular disease. Can we take this a step further into brain activity and brain disease as well? Microplatelets are derived from megakaryocytes in the bone marrow, which have a nucleus, and therefore they'll be responsive to hormones. Their activation and production of microvesicles will affect the capillaries and the vascular wall to increase the development of lesions and perhaps change capillary activation and permeability. This will affect end organ structure and function, and hormonal environment will influence this because of the genomic effects of the hormones on the megakaryocytes. The microvesicles are derived from activated cells, but we need to consider what might activate these platelets in the circulation. First of all, we have agonist-stimulated activation of the platelets, but also they are activated by things like systemic blood pressure, fasting blood glucose, triglycerides, and inflammatory cytokines. These are conventional cardiovascular risk factors which change with sex and over time with aging. They're also influenced by pregnancy disorders and menopause. So we have the sex-specific conditions also affecting either directly or indirectly platelet activation, thrombin generation, vascular structure and function. What actually are these microvesicles? They're cellular membranes which carry signature markers of the cell of origin. They also provide a surface for the generation of thrombin, and they contain bioactive molecules which, when adhered to a cell surface, can release their cargo, affecting subsequent target cell activity. We evaluated the association of white matter hyperintensity with the platelet activation in the women in our Cronus early estrogen prevention study. Again, these women were within the age range 42 to 58 when they came into the study, and after the study was finished, some of them were 62. We looked at the platelet characteristics of these women when they came into the study, that is at baseline, and found that the expression of fibrinogen receptors correlated to the white matter hyperintensities at the time they came into the study. This reflects their potential thrombin-generating activity. We also looked 4 years later at the white matter hyperintensities and found that these correlated with the platelet-derived microvesicles, not at the time we imaged them, but 4 years before, which tells us that perhaps there is a temporal sequence, a time-dependent factor of when cells become activated in the blood, the time it takes for them to interact with the vascular wall, and actual structural changes in the end organ. So how do we wrap this up into the big pictures that we've learned over these studies? That sex does matter. We need to pay attention to it in every experiment, and I would challenge you as you go forward today to listen to the subsequent speakers, that if they actually talk about the sex of the material they're investigating. We've heard about taking an integrative approach. I think we need to take an integrative approach to causality of small vessel disease, thinking about how changes in the kidney affect the brain and the heart, and actually consider the temporal sequences of the events that we're studying, that what markers we're measuring today may not indicate the factors which are happening in that organ we're imaging, but going forward. Finally, as we go forward, I think we really need to better define what we mean by small vessel disease. We need to figure out, excuse me, that when sex matters and how it matters. And finally, is small vessel disease a continuum of large vessel disease, or is it an independent entity? And how is the feedback worked out? And finally, we need to develop better diagnostic and prognostic tests and markers to indicate when small vessel disease occurs and the subsequent changes over time. Thank you very much.
Thank you, Virginia. Any questions?
Yeah, Paul Nyquist from Johns Hopkins. I just want to congratulate you on your great work, and I would like to discuss not only the issue of white matter progression within women, but in specific brain regions. And there's this new terminology that has come up about subcortical structures and classifying them as deep white matter hyperintensities versus periventricular hyperintensities. And there's been a couple small papers and longitudinal studies that show that in female subsets there's greater expansion of the deep white matter hypervolume. We've also now seen this in our study, where we've had 800 individuals, and there's a strong association that is independent of age, suggesting that deep white matter hyperintensities at periventricular are not only independent from age, but that deep white matter hyperintensities are strongly associated with female sex.
Thank you very much for that comment. We tried to evaluate our white matter hyperintensities with brain regions in the first study we did. Our imaging technique was not on, I'm telling you what my radiologists tell me, okay? The technique that we used and the machine that we used in the initial study was not sensitive enough for us to be able to break down in the areas of the brain that we would like to see. As we're calling these women back 3 years after they came out of KEEPS, we're using a different machine that's going to allow us to better analyze the subcortical areas where where these white matters may occur. It's a very important question. We understand it. I'm glad you're finding it, and I'll have to talk to you later about exactly how we can go forward.
Great. You're working with Dr. Jack, is that correct?
Yeah.
Mm-hmm. Yeah. I think he's working on this issue aggressively now.
Yeah, I know. That's why we've changed some of the techniques that we're using to image the women coming forward. And actually, the pregnancy cohort the epidemiology study that I mentioned as part of our SCORE project, we're actually calling these women back 30-some years or 20-some years after they've had their pregnancies, and we're giving them a complete cardiovascular workup, as well as cognitive function and brain imaging as well. So we'll really be able to understand within these women that had a hypertensive pregnancy disorder what the subsequent cardiovascular and cognitive challenges are for these women.
Well, thanks for your work. It sounds like a very important clue. So.
Please keep your questions very short. I think we can only take one more. We have a panel coming up.
Okay. Zina Wexler, University of California, San Francisco. A quick question. Microvesicles, thrombin-containing microvesicles, are those exosomes or they are microvesicles?
Okay, we defined our microvesicles in terms of size, so it's anything less than 200 micrometers. And we have specific, what's hard is to separate out markers from microvesicles and exosomes because some of them still carry the same kinds of markers. We're working on that now with different types of centrifugation and gradient separation to try to find the exosomes. What we have identified in this study were those microvesicles that were defined by having an Nexin 5 positive carriers, P-selectin, endothelial markers, and platelet markers. We haven't in this study separated out the exosomes, but we're working on that when we're looking at the products in the urine that may be affected subsequent by kidney disease to really pull out the exosomes, the internal organelles of the cell compared to those of the cell surface.
Thank you.
Right. Thank you very much.